going to talk a bit about virus vocabulary. So I'm just going to go over some terms that are going to be useful for the rest of the video. So a capsid. So we're going to be talking a bit about a capsid. And a capsid is just a protein protective layer that surrounds the genetic material of the virus. So made of proteins, and it's basically just protecting that genetic material. We're going to be then talking a bit about viral specificity. And viral specificity is just how certain viruses are very specific to a given host cell. So, for example, there's certain human viruses that only infect certain types of human cells. They will not infect other animals, and they may not even infect other types of human cells. Then we're going to be talking about two types of viral reproduction. The first type is called the Lytic cycle, and the other type is called the Lysogenic cycle. And I'm going to go into more detail on those later in the video. So now, we were just previously talking about the criteria of living organisms, so how we classify something as living versus non-living. And viruses are actually not considered to be alive based on these criteria. So remember, if we go back, our first two criteria here was that, one, all living things need to be made of cells. And as we're going to talk about, viruses are actually really not made of cells. So they do not have a membrane-like structure like all cells have. And the second part is that viruses cannot reproduce independently, so they cannot reproduce on their own. And we're going to be talking about that when we get into the lytic and the lysogenic cycle. And essentially, viruses need some kind of host cell to be able to carry out their reproductive cycles. Alright, so now let's go into the structure of viruses. So there's a huge diversity in the shapes and kind of sizes of viruses that are out there, but they all have some things in common. So generally, viruses are only made up of two types of biochemicals. So these two types of biochemicals are nucleic acids. So remember, nucleic acids can be, for example, like DNA. So DNA is one type of nucleic acid. Another type is RNA. So we've talked about both types of these nucleic acids in our genetics unit, so it should just be a recap for you. And the other type of biochemical that viruses are made of are proteins. So remember, proteins are the essential building blocks of life, and they are made up of amino acids. So proteins form a protective shell around the DNA, or sometimes RNA, and this protective shell is called a capsid. And this is just something I want to highlight. A lot of viruses have DNA as their genetic material, but there are a lot of viruses as well that have RNA as their genetic material. I'm going to now give an example of a type of virus that actually infects bacteria. So this virus is called a bacteriophage. And bacteriophage literally means bacteria, so bacteriophage, which means killer. So bacteriophage is something that kills bacteria. So this particular bacteriophage actually infects E. coli, and E. coli is a bacteria that is found in our own gut and actually helps us digest food. So if we look at this diagram on the left here, you'll notice that in black, this is all protein. And we have the capsid. This region here would be considered the capsid. So it's just a protective protein kind of shell that surrounds this genetic material within the capsid. And in this particular virus, the bacteriophage T4, the genetic material is DNA. We can also kind of break this bacteriophage down into different body parts, if you will, and we can consider this section to be the head, this section here to be the collar, and this section down here to be what's called the tail. And it's this tail region that will actually attach onto a bacteria cell, and the genetic material will then be injected into the bacteria cell from this tail region. Now I want to just kind of give you a bit of an overview of the diversity of viruses that are out there. So we've just talked about bacteriophages. So here is actually an example of a bunch of bacteriophages attacking an E. coli cell. So you can see here, this top part here in white is actually the capsid, and so it's this head region of the virus. And then these kind of spindly-like structures here are actually the tail regions, which are attaching to this bacteria cell, and then they're going to be injecting their DNA into this bacteria. So some viruses have this similar kind of alien-like structure that we talked about in the bacteriophages, but some can have more of a spherical shape. So this particular virus has more of a spherical shape, and it has these kind of projections coming off them, which are actually just protein projections. And so as you can see, there's quite a diversity in the total shape of viruses that are out there. This particular example down here is actually a virus that infects humans and can cause tumors to grow. And the reason that I included it is because you'll notice that there's actually a line across each of these viruses, and that's just showing that the diameter of these is all around 50 nanometers. And a nanometer is a very, very small unit of measurement. So 1 nanometer is equal to 1 billionth of a meter. So a very small unit of measurement. And you may have kind of been wondering, why do I have all these pictures in black and white? Why don't I have any color images of viruses? And that's because we actually don't have any color or true color images of viruses, because all the images we have are actually taken with an electron microscope. So these are all pictures taken from an EM or an electron microscope, and there are no color images from electron microscopes. And bacteria were actually discovered a lot earlier than viruses were, because the traditional light microscopes that scientists were using early on were capable of seeing bacteria, but they were not powerful enough to actually be able to see viruses because they are so small. So it wasn't until the electron microscope was invented halfway through the 20th century where we actually were able to start seeing viruses. Now I'm going to talk about viral specificity. So for a virus to enter a cell, proteins on the outer surface of the virus must match those on the outside of the cell that it's going to infect. This means that a given virus can only infect a very specific type of cell. So hence the term specificity. Certain viruses can only infect certain types of host cells. Now I want to talk about viral reproduction. So remember that viruses cannot reproduce on their own. They require a host cell to reproduce. There are two main patterns of viral reproduction or infection, and we can use the term infection because viral reproduction is essentially just infection. So there is the Lytic cycle and there is the Lysogenic cycle. So first I'm going to talk about the Lytic cycle. So the Lytic cycle is when a virus will infect a host cell. So this example here, we're actually using a bacteriophage like we just talked about, and a bacteria. So this bacteriophage is injecting its genetic material into this host cell. And this genetic material will actually be separate from the original host chromosome here, or host genetic material. So it's separate from the genetic material of the host. What's going on in this first step here is that the genetic material of the virus is being mass replicated. So we're undergoing DNA or RNA replication, and it's getting mass replicated so that we're getting a lot of copies made of the genetic material of the virus. And this is using the machinery of the host cell to do this. What we're getting now is, in the next step, is there is going to be proteins made from that genetic material of the virus. So the template that's being used is the DNA from the virus cell, but it's using, again, the cellular machinery, so like the ribosomes of the host cell, to create those viral proteins. And these viral proteins can self-assemble into different parts of the virus. And these parts of the virus are then going to form full new virus particles. And once the cell is so full of new viruses, it eventually will lyse or split open. And those new viruses will be released into the external environment of what the cell was in, and could then go on to infect new cells and start this lytic cycle again. So that's the lytic cycle in a nutshell. And now I'm going to go on to the lysogenic cycle. So some viruses just have the lytic cycle, where other viruses have the lysogenic cycle and then later go into the lytic cycle. So we're going to start here for the lysogenic cycle. So we're going to start on this one again. And we have the virus which is injecting its genetic material into the host cell. But what you'll notice here is that the virus genetic material, which is shown in red, is actually being incorporated into the genetic material of the host cell. So what's happening is that the virus is kind of lying dormant throughout this lysogenic cycle. So when the host cell's genetic material is replicating, as shown here, the virus's genetic material is going to get replicated kind of just hanging along with it. And what this is going to do is it's going to create a lot of copies of the virus, but without actually infecting or damaging the cell as of yet. But eventually, this is going to go into the lytic cycle. So from the lysogenic cycle, we're going to go into the lytic cycle. So once there has been many copies and the cell has divided a number of times, now there's multiple cells that have this viral DNA, they're going to go into the lytic cycle and go through the same steps we just talked about. So the viral DNA is going to get reproduced, viral proteins are going to be created, new viruses are going to get assembled, and finally we're going to get the lysis of cells and the release of new virus particles again. So this was the lysogenic cycle, which is then followed by the lytic cycle in certain types of viruses. So now I just want to recap the main points from today's video. So there were 5 criteria needed for an organism to be considered living. We covered the basic structure of the bacteriophage T4. We talked about how viruses have viral specificity, meaning they can only infect a specific type of host cell. And we talked about the 2 main patterns of viral reproduction or infection, which were the lytic cycle and the lysogenic cycle. Thank you very much for watching this first episode of Hollick Kenyon Science. Hope to see you again soon.